Hey guys, here's your overview video of chapter 15. Prokaryotes and eukaryotes can alter gene expression in response to their environments. So this is a really useful function for organisms to have, and gene expression is often regulated at the transcription stage. So that's what we're going to focus on today with gene regulation, is somehow altering that transcription part, the part where DNA is made into RNA before it makes a protein. So we're going to talk about those controls. And this has been favored by natural selection because then cells aren't wasting energy producing stuff that isn't needed by the cell. It only produces it when it needs it. And we're going to talk about operons. So gene expression in bacteria or prokaryotes is controlled by operons. Eukaryotes, we do not have operons. We use a different type of regulation, and we'll talk about that as we go. So here's just the two types of regulation. We can have regulation by enzyme activity. So remember this feedback inhibition. When we get enough of this, then there's a feedback loop that prevents this from continuing to happen. So prevents the product from continuing to be made once we have enough. There's also regulation of gene expression where this protein isn't made from DNA when there's enough. So this is the side that we're going to talk about today. All right, operons is a whole stretch of DNA that includes an operator, a promoter, and the genes that it controls. This is, again, just in prokaryotes. That operator piece is the segment of DNA within the promoter, and this is the switch that controls the access of RNA polymerase to the genes. So the operator is what allows transcription to occur or not. The operon is really useful because everything is all together. So it can either happen, transcription can happen, the switch is on, or it can be blocked, and that means the switch is off. All right, repressors can bind to the operator, and that blocks transcription from occurring. Repressors are made by a regulatory gene. Co-repressors can also function, and we'll talk about those as we go. All right, two main types of operons. Again, this is just in prokaryotes or bacteria. So a repressible operon is usually on, but it can be repressed, meaning it can be turned off. And an example of this is the tryptophan operon. An inducible operon means we can induce it to turn on. So it's usually off, but we can turn it on. And an example of this is the lac operator. So by turning it on, typically it has this repressor bound because it's usually off. A molecule can bind to the repressor that can make the repressor not function, and that will turn it on. All right, so we are going to talk about the two types. So tryptophan operon usually is not present in the environment, so E. coli has to always make it. So that means the tryptophan operon is always on, and the genes for tryptophan synthesis are transcribed. But let's say tryptophan happens to be present in the environment, the cell doesn't have to make it, so that's when the operon will be turned off because it doesn't need to make it if it's in the environment. So here is the picture. So this is when tryptophan is absent. Here's our repressor. So you can see here's the regulatory gene that makes this repressor. When tryptophan is absent, the repressor is inactive and the operon is on. So the cell needs to make the tryptophan. So here is the whole operon here. You can see here's the promoter, including the operator. This is the region where RNA polymerase is going to bind, and then it's going to transcribe all these genes of the operon. These genes for the operon then get transcribed into mRNA, and then these mRNA gets translated into the polypeptide that makes enzymes for tryptophan synthesis. So this is when tryptophan is not present in the environment. When tryptophan is present, the repressor is active, and then the operon gets shut off. So you can see when tryptophan is present, it actually binds to this repressor and activates the repressor. So this repressor comes in and binds to the operator. So you can see RNA polymerase can't bind now, so transcription does not occur. All right, so that's the tryptophan operator. The lac operon is inducible. and it contains genes that code for enzymes used in the metabolism of lactose. So it breaks down lactose. So by itself, the LAC repressor is active and it switches the LAC operon off. So a molecule called an inducer inactivates the repressor. All right, so lactose is absent and the repressor is active, so the operon is off. So this is the normal state where this is off, right? So it's inducible, meaning it's usually off, but it can be turned on. So here, when this repressor is made, it's made and in the active form, so then it just goes ahead and binds to the operator. RNA polymerase cannot bind, so no RNA is made. Now when lactose is present, this allolactose acts as an inducer, binds to the repressor, and so now we have an inactive repressor. So the repressor is not on the operon anymore. RNA polymerase can bind and transcription can occur. So then all of these enzymes to break down lactose that's now in the environment are made. So inducible enzymes usually function in catabolic pathways, meaning we're breaking something down. And repressible enzymes usually function in an anabolic pathway where something is needed to be made. So inducible, you can see, is here where lactose is present and it's being broken down. Repressible, remember, was the tryptophan, which is tryptophan is being made. So this is repressible, meaning it's usually on, but it can be turned off. Both of these together are considered to be, so tryptophan and the LAC operons are both negative control of genes because they can be switched on and off. So the off means that it's a negative control. There's also positive gene regulation. So this is by CAP. So CAP is a catabolite activator protein. So it acts as an activator. It's activated by binding with cyclic AMP. This should sound a little familiar. This activated CAP attaches to the promoter and increases RNA polymerase and accelerates transcription. So it's positive. This positive gene regulation increases the rate. So it's just this extra thing on the promoter that's kind of waving down RNA polymerase, being like, hey, over here, come bind, which then helps the process happen faster. So both positive and negative can happen at the same time with the LAC operon. So the LAC operon has negative control by the LAC repressor, and then it also has this positive control by CAP. So here's the picture of the positive control. So when lactose is present and glucose is scarce, that's when the cAMP levels are high. So that's when this happens. So you can see this cAMP binds and creates this active cap. This comes on here, and then RNA polymerase binds. This is like an extra flag or signal for RNA polymerase, and then this increases the production of the enzymes. All right, so here's when lactose is present and glucose is present. So this means the cAMP levels are low, so that the positive control does not happen and CAP remains inactive. So this remains inactive. RNA polymerase is less likely to bind, but it still can because that LAC repressor is not on there. So this still gets transcribed, just not as quickly as when we have this active cap there. All right, so that was prokaryotic gene regulation. We're going to move into eukaryotic gene regulation. So this one, there's different stages. Remember, eukaryotes do not have operons. So most regulation occurs during transcription as well. But here are all the different points in which regulation can occur. So we're first going to talk about chromatin modification. So remember, chromatin is like the chromosomes, but remember, it's just when they're not condensed. So the chromatin is just kind of like a jumble of all the DNA. And what can happen there is the structure of the chromatin can be changed. So what can happen is acetylation, and this promotes transcription by loosening the chromatin structure. So when acetyl groups are added, so you can see here how there's just these spikes coming off, Here, acetylated histones are present, so that just means these are added to the histone tails acetyl groups, and that unwinds the DNA. So it loosens the chromatin structure, which then makes it easier for RNA polymerase to come in and bind and for transcription to occur. Opposite is DNA methylation. So this is when methyl groups are added, and this condenses chromatin and reduces transcription from occurring. So both of those modifications are kind of called epigenetics. It does not alter the DNA, right? The DNA is not changed. And this can be passed to future generations, but this is called epigenetics. So epigenetic inheritance can occur, but these can be reversed. All right, so moving on. So there are control elements within transcription as well. So there's enhancers, which are distal control elements, meaning they're farther upstream, they're farther away from the genes that are going to be transcribed. And then there are proximal control elements too that are right before the promoter. So you can see here's those. The enhancer is located upstream, and then here's those proximal control elements. All right. So to initiate transcription, eukaryotic RNA polymerase requires the assistance of transcription factors, and those proximal control elements that are close also help, as well as the enhancers, those distal control elements. An activator binds to the enhancer, and that helps protein-to-protein interactions. So here is an activation domain, and you can see this kind of all comes together. The bound activators are brought into contact with mediator proteins through DNA bending, and those mediator proteins interact with proteins at the promoter. So I'll show you a picture. So this is what it's going to look like. So here is the segment of DNA that's going to be transcribed. Here's RNA polymerase, and then we have all these other things here. The DNA actually bends, so here's a bending protein. So that way, this distal control element, this enhancer, then we have the mediator proteins. All of this comes together as like this complex to help RNA polymerase transcribe DNA. So an activator binds to the enhancer. DNA bending protein comes in and bends the DNA so that this enhancer region is right over the promoter. Then we have this group of mediator proteins and general transcription factors that come in to kind of make a bridge. So you can see here's the bridge between the enhancer and the promoter, and then RNA polymerase comes in and binds. So again, this just helps. It signals RNA polymerase that this is the spot that it should be transcribing. All right, some transcription factors can be repressors, but mostly we have activators that help transcription to happen. But sometimes there are repressors that would block transcription. So you can see this is part of the reason why different genes are expressed in different cells. So you can see because every cell has the exact same DNA, right? So it just depends on what activators are available. So you can see in a liver cell nucleus, we have different activators that are in a lens cell or a cell in the eye. So see the different colors that shows that there's different activators. So depending on what's there, those different activators will bind to the enhancer, and then that will create a different product. So here the albumin gene is expressed. Here it's not expressed because those enhancers are not present. A crystalline gene is expressed instead. All right, so genes can be scattered over different chromosomes that are needed at the same time, but each one has the same combination of those control elements, so transcription can happen at the same time. So it's like a mailman looking for flags up on the mailbox. All the cells have those activators and those same control elements. And so those activators can bind and transcription can occur at the same time, even if it's on different chromosomes. Alternative RNA splicing is important as well. We already talked about that, but different mRNA can be produced from the same transcript. and that happens here. So we talked about chromatin modification already, transcription, and then RNA processing. All of these are ways that genes can be regulated. mRNA can also be degraded. So this is another way that regulation can occur. And then after translation, regulation can happen by other techniques or ways as well. I forgot to talk about this part. So there are non-coding RNAs. They're little tiny pieces. So microRNAs are small RNA molecules that can bind to mRNA sequences. So this would happen after transcription. And then they can degrade or block translation. So mRNA can be degraded, which then means a protein will not be made. Or it can also inhibit translation from happening. So you can see here's this miRNA, this little RNA that's made. It can bind and then translation will be blocked. Or mRNA can be degraded as well. Interfering RNAs are just other small RNA that can also cause RNA interference. All right, just a few other genetic engineering things. So RT-PCR is reverse transcriptase polymerase chain reaction, a little hard to say, and it turns mRNA into DNA. So it basically works backwards. So if for some reason we have mRNA, we can actually amplify that once mRNA can be made into DNA, and then that can be amplified, and then we can actually do gel electrophoresis based off of that. All right, so different types of regulation, gene regulation in eukaryotes. We can have chromatin modification, RNA processing, where we're using that alternative RNA splicing. mRNA gets degraded. We could edit, or I'm having trouble with words right now, so we're almost done. Stick with me. Translation may or may not occur, and then protein can be processed in different ways as well. So those are all the different ways that eukaryotic genes can be regulated. That is for class time. All right, hope that helps. Bye.